talked a lot about APR. I think Robert explained it very well. These are actual pictures. From my experience, that's what the kingbird seedling infection type looks like. It's not a complete 4. It's a 3+, 3++. But it seldomly goes any higher than this in the field. Low MR, MS response. Again, another example, susceptibility versus MR for stem rust. And quite easy to pick if you have, again, if you have a good reference point in your field plots. So let's see how you go with the scoring. What would you call that? That comes from the field. It's not a greenhouse grown. Yeah. You can see the purple stem color. The stem melanosis or melanin pigment is linked to the SR2 gene. So that's quite easy, I think. That's a 0R. No visible disease signs. And we talk about symptoms, but actually we should talk about signs. Symptoms are mottling and things like that, or dwarfing, or signs are the actual sporulation and the pustules, etc., etc. All right, next one. Small pustules. That's not susceptible, is it? It's also R. Now it gets a little bit more ugly. Some larger pustules. What would you call that? MR? I would call that MR too. You can even call it R2MR because you see quite a number of R's in there and then some MRs. Not a very high severity, few large pustules. I would call that MR to MS, yes, or then just M, if you want to use the, I think Simmer just uses M. MS. I'd also call that MR to MS, but the high severities are probably 60 or 70 MR MS. MS? Yeah, when they're chronologically ordered, so it's easy. But in the end, they're not, so when we get to this, the last part. This is an interesting one. It probably needs a better description in the literature, I think. What's the gene involved here? SR2. Yeah, SR2. But if you look at this part, that's almost 100S on that stem section. But in the context of the entire stem, it's not that high. So I think we need to explain this if we take the notes in the field. And I would say MSS, or and then just node and make a, if I say node in my book, my field book, I will know it's a SR2 type of response. We get a distribution of the susceptible ones just above the node, and then the rest would be resistant. It's very typical of APR in many cases.
Do we know why that is?
I don't know. I think I've read that there's a concentration of nutrients in that particular part of the stem, but I don't know whether it's true. I've always wanted to make sections and then compare the histology. But I may stop rust work before I get to that point.
I think anatomically, if we look into the anatomy or histology of the cross sections of this node area, there are more safe tubes in here.
It could be.
Usually. And when there are safe tubes, transportation of certain nutrients across these tubes, it's limited. And that's why we see more infection in the above areas and less in the down areas. That's my perception, maybe I'm wrong.
Yeah, I think there are still things we can learn about stem rust, although we have many, many papers on this particular disease. I think that's fairly clear. MS? You agree? Even MS to S. MS to S probably, I don't know what I got there coming up. MSS, yes. And then finally, a nasty one. You can also do this on barley if you use other crops. I just put in a few slides. The reaction types are slightly different, but you get the same variation from resistant, moderately resistant, all the way up to susceptibility. I hardly ever see 100 S's on barley, but you see the susceptible response time. Just another nice example of the SR2 gene linkage. There's another one. Again, a large pustule just above the node. The rest completely resistant almost. There's one was fairly susceptible. And I think we've also heard about the SR2 complex. When you have the SR2 complex or you have that combined with major genes or all-stage resistance, you'll get a clean plant. But when you only have SR2, infection levels can go pretty high. And that's one of the questions Gordon wants to answer. What protection is actually given by APR? Different levels of APR. Just again, we've also, I think, the LR34 linked leaf tip necrosis was mentioned previously. In our conditions, it could be very severe. And I think the dead leaf tips may also contribute to leaf or to yield loss rather than the disease itself. What do you see on this leaf?
X.
You see an X here, but can you be more specific? You assume that it's all stem rust. There's leaf rust there as well, yeah. So make sure when you take the notes, usually stem rust does not occur on the leaves, but you can see it under very heavy infections. That makes sure that you can distinguish between leaf rust and stem rust pustules. We move on to stripe rust. I think most of you have experience, probably more experience with this, and the susceptible versus resistant phenotypes. And I'm not going to go through these one by one as we did with the stem rust, but we usually have a resistant range. We have a fairly large intermediate range, MR to MS, and different combinations, and then MS to S. There are quite a few of these slow rusting genes, adult plant resistance genes in stripe rust. If you postpone your notes to the end of the season, you won't see them. But if you calculate disease progress curves, or at least draw your disease progress curves, you will see that at the early stages, you will get plants that look like this, when some of the others are already there. So very important to get your timing right as well. These are not easy to score, let me tell you. If you really want to be detailed and specific, and you can spend a lot of time and waste a lot of time arguing with yourself or your colleagues. Now, is that MR or MS? And do we see necrosis and do we see sclerosis? So I know some of the people with large programs, they would just score it as M, or they will even leave out the response type and just score percentage leaf damage.
We use 0 to 9 scale.
You can use a qualitative scale as well,
Yeah.
ordinal scale.
Sometimes it depends on what the material is you're storing.
Exactly.
So for us, if it's very early generation material, breeders, they just want to know whether it's any good or not. And 0 to 9, we can run through very quickly and cull material based on that. If it's segregating genetic material.
If it's a mapping population, you'll spend a lot more time. But again, don't waste your time to decide whether it's 43.75% or 42.12%. You've got to be clear in how you score. And we did an experiment during the past 2 seasons. I have reported some of this at previous meetings where we used remote sensing or close-range remote sensing using infrared and near-infrared. You can buy your green seeker device commercially. And we wanted to see whether we can actually use this for stripe rust. So you would angle it towards your row. Again, the textbook tells you that the device should be held over the plot, above the plot, but again, you can see all the awns and the spikes are still green, so that they interfere with the reading that you get. So we figured out that when you put it at that angle, about 50 centimeters from the row, and you pull the trigger, you get a very good response. And there you can see some of my colleagues doing this in the field, and it's been published that it works. That's not new, but what we've done now is we have actually used the green seeker scores as a trait in mapping QTLs in populations for which we have genetic maps. And in three populations, we came up with exactly the same QTL. So they're very, I think, handy. It takes you about 20 minutes to go through a few hundred of those. There are things to keep in mind, but it can work. So that's just the point I want to make.